Hi everyone, today I'm going to talk about the final project. We developed a computational method for doublet detection in single-cell RNA-seq dataset via the semi-supervised adversarial autoencoder. Our group member is Andy Chu, Yuhang Chen, and Doublet. So a little bit introduction and motivation. In single-cell RNA-seq protocols, one or more cells could be assigned one single barcode, as shown in this diagram, which are named doublets, and this will lead to some confounding results in downstream data analysis. Some experimental techniques for doublet detection, including cell hashing and demoxlet, have been developed. However, they suffer from special instrument and expensive costs. So our goal is to detect and remove doublets from unlabeled single-cell RNA-seq dataset, and there are several computational approaches to do this. Most of the computational approach generate artificial doublet and then compute the similarity between artificial and true doublet. The key difference, however, is that they use different algorithms for computing the similarity measurement. Despite the progress of developing computational approach for doublet detection, there are some limitations. One of the limitations is that the accuracy is not high. The best detection method, doublet finder, only achieve a mean AUPRC value of 0.537. The accuracy is also not consistent across methods, and also performance under distributed computing decreased due to a loss in global information. So inspired by recent work on variational autoencoder-based doublet detection method, we adopt an adversarial autoencoder method for doublet detection. So an AAE combines the design of a VAE and a GAN with the rationale that an AAE is more sensitive to outliers as compared to regular GANs. So we hypothesized that an AAE could generate enhanced single-cell RNA-seq expression profiles to detect outliers, which are doublet in our case. So for a classical VAE, here's the loss function. For AAE, the KO divergence is replaced by GAN, which the latent vector is then fed to a discriminator. So here is the architecture of our model. So the input data is fed to an encoder to learn the compressed representation Z in the latent space, where Z is forced to conform to a normal distribution. The latent coding Z has three downstream usage. First, the latent Z is passed to a decoder to reconstruct the data input. Second, the latent Z and the normal samples from a normal distribution are passed to the discriminator. Last, the latent Z is passed to a two-layer dense neural network classifier to predict whether it represents singlets or doublets. So our results consist of three parts. The first part, we compare the generated doublet and real doublet. The second part, we're trying to see if the latent variables produced by the AE are close to the prior distribution, which is Gaussian distribution. In the last part, we try to test the performance of our model to see if the model can correctly classify singlets and doublets. So as you can see from this figure, the generated doublets and real doublets are very similar, but they show significant difference from real singlet, which indicate that the generated fake doublet are acceptable for subsequent data analysis. So next, we concatenate the generated fake doublet to our real data set, and then we split into a training and testing set with a ratio of 4:1. The best scenario is achieved, which composed of 3 two-layer network. So here it shows a diagram of the 8 latent variables after training for 100 epoch. The latent space is chosen to a normal distribution for most of the dimensions, which indicate that the GAN structure successfully forced the latent distribution to align with the prior distribution. Next, we want to test the reconstruction error among real singlet, real doublet, and fake doublet. As we can see from this table, the reconstruction error of doublet is generally larger than the real singlet, and this result supports our hypothesis that the doublet will behave more like outliers when generating the Gaussian latent embedding. So next, we test the performance of the classifier. So the left figure shows the real singlets, the real doublets are both labeled and singlet. matching the labeling during the training, and the corresponding AUC is 0.88. The right figure shows that the label of the real doublet is flipped to doublets, which align with the ground truth, and the corresponding AUC increased to 0.9. This result suggests that the model intended to label the real doublet as doublet, even though those cells were labeled as singlets when training the model. So the conclusion is that we built a semi-supervised approach for doublet detection, which consists of an AE and a two-layer classifier. Our model successfully learns a low-dimensional embedding and achieves promising accuracy for doublet detection. Some possible future improvement including hyperparameter tuning, biological downstream analysis on how generated doublet differ from real doublet, and also downstream biological analysis for the embedding layer. And thank you so much for listening.